Hello, my name is Anthony Perez, and I will be presenting the article titled Effect of Microalgae Application on Soil Algal Species Diversity, Cation Exchange Capacity, and Organic Matter After Herbicide Treatments. Starting first with agroecosystems, which are agricultural units managed by people. These units are also governed by interactions between living and non-living factors, and like in natural systems, experience disturbance events. In the case of agroecosystems, disturbance events would also include human activities, both intentional and unintentional, such as tillage of the land in preparation for crop planting and herbicide applications to suppress weeds, and many other agricultural operations. Which brings us now to soil microbes and the positive effects that they can have on soil structure and health in terms of bringing the soil together and contributing organic materials for other members in the soil community to utilize. Microalgae in the soil are a significant biotic factor in the environment and contributor to soil ecosystem services. However, the extent of the role that they play after continuous disturbance events in an agroecosystem are not well understood. Another important ecosystem service that microalgae provide is by acting as indicators of soil health, which is observed by parameters in the soil such as cation exchange capacity, percentages of organic matter, pH values, and species diversity. Thus, the researchers of this study sought to establish a correlation between artificially increasing microalgae populations in the soil with effects on soil parameters such as percent organic matter and cation exchange capacity across soils of both low and high pH values. The second objective of this study was to track changes in algal species diversity by comparing plots separated into three categories: the control, plots with high pH, and plots with a low pH. The materials and methods used by the authors of this study included significant pre-preparation of 5 half-acre plots that would later grow corn or Z maize, and the significant preparation of the soil was necessary before the official experimental treatment of Go Green, which is the trademark name for the microalgae application. Pre-preparation of the soil bed consisted of strip tillage along with fertilizer application in the month of April, a second fertilizer application in the month of May, and a third using sprinkler irrigation in June and July. Herbicide applications began in May and ended in June and used 4 different products, including Degree Extra, Roundup, Roundup Weather Max, and AMS 17. Go Green applications began in June and were applied through center pivot irrigation to plots 1 through 4, where plot 5 was the control. Interestingly, plot 3 was chosen to study the effects of consecutive years of planting and harvesting on the previously mentioned soil parameters and algal species diversity counts. Each plot was further separated based on high and low pH values at 8.3 and 6.8 respectively, and will be referred to as H and L plots for the purposes of this poster. Soil samples comparing the changes to parameters in soil such as CEC, percent organic matter, and pH were taken after herbicide treatment, at the time of harvest, and 2 months after harvest. The samples measuring algal diversity were collected somewhat differently, where sampling occurred in relation to the vegetative and reproductive stages of corn crop development and occurring in 3 separate instances at the V5 stage, the V6 through 11 stages, and the final R6 phase, which represents the corn's physiological maturity. These samples compared differing counts in blue-green algae, green algae, and diatom groups. These groups were cultured using a combination of selective media, a technique adapted from the 1950s that uses soil water to obtain pure cultures, and the spread plate method on solid media to obtain single colonies. Light microscopy techniques were then used to examine the morphological differences between the 3 groups. The authors then used 2-tailed paired difference t-tests in order to compare the groups before and after treatment of Go Green to determine any significant effects on the previously mentioned soil parameters. This brings us to the results of the study, which in almost every case of tested soil parameters, there is found to be significant differences in each plot treatment and pH group. Let's look first at the pH differences in soil before and after treatment of Go Green, and are shown in figure 3. We see that the pH values in both L and H plots generally began with higher value post-treatment of go-green. Noticeable changes in L plots with a pH over 7 include a sort of stabilizing effect in pH closer to neutral 2 months after harvest. Although generally for both L and H plots 2 months after harvest, pH values did decrease with the exception of plots 3 and 4 for H plots. The same stabilizing trend seen in L plots to reach a neutral pH was not seen in H plots 2 months after harvest. These plots actually showed a 0.2 unit increase, contradicting the data seen in H plots 1 and 2. And it is interesting to note that plot 3 was the consecutively tested plot, and plot 4 included applications of GrowGreen at twice the rate of the manufacturer's suggestion. In the end, there was significant difference, or p-values, of less than 0.05 between L plots at the time of harvest and 2 months after harvest. Differences in organic matter percent changes were also significant, where L plots showed lower values than the control, although increased with treatment in 2 months after harvest, which contrasts with data shown in H plots, where a decrease in organic matter was observed 2 months after harvest, including the control. Controls of both L and H plots so differed in that in L plots, organic matter values didn't change over time, whereas in H plots, a decrease was observed 2 months after harvest, indicating factors other than GrowGreen application were related to this decrease in organic matter at this high pH range. In the end, both L and H plots showed a significant difference in treatment at harvest and 2 months after, with p-values less than 0.05. Next, differences in CEC, or cation exchange capacity, were observed in both L and H plots at harvest and 2 months after, and are illustrated in Figure 4. It can be seen that in L plots 1, 3, and 4, which had the highest applications of GrowGreen, the largest increases in C and C were observed, and a uniform decrease in all plots, including the control, was observed 2 months after harvest, which more or less aligns with the draining effect in the soil after plant uptake of cations. On the other hand, in H plots 2 months after harvest, the same uniformity in value between plots was not observed. Additionally, CEC values for H plots 2 months after harvest were also lower, even though H plots, in comparison to L plots prior to harvest, were significantly higher. Between L and H plots at harvest and 2 months after, significant differences, or p-values with less than 0.05, were observed. Lastly, it is interesting to note that for L plots, average values of the control were 0.8 units lower than the average value for all plots in that same pH group, reflecting these plots' greater ability to retain nutrients and ions and demonstrate the buffering capacity of CEC. In relation to algal species diversity, total populations of blue-green algae were 60%, green algae at 25%, and diatoms at 15%. In L plots or soils with pHs less than 8.0, green algae were more abundant, which correlated with the control at that pH range, compared with blue-green algae at pH ranges above 8.0, green algae were lower. At the lower pH range, diatoms increased after treatment compared against the control. Unlike the other tested soil parameters, comparison of species diversity for algae made use of total counts of the 3 algae groups across all plots, but separated still by pH ranges, where the total counts following treatment of GrowGreen 2 months after harvest resulted in a 50% green algae, 5% diatoms, and 45% blue-green algae, meaning that blue-green and green algae counts were similar to counts prior to treatment. And regarding the controls, blue-green algae populations dropped by 50% when compared against green algae, as can be seen in Figure 6H. The indication of differences in microalgae diversity can be seen when viewing the low and high range controls and noting their small differences, and comparing the populations of these two same groups in treated plots, suggesting diversity counts are based on pH. The ratio of percentages in blue, green, and green algae counts were higher than those in control plots, supporting the effect of treatment on algae populations. And over time, green algae counts were higher than those of blue-green algae when comparing treated versus control plots. Results obtained in the study showed significant effects from GrowGreen applications on the previously mentioned soil parameters, as well as favorable accounts for algal species diversity, and when taken together suggest positive effect on soil formation. Although biotic and abiotic factors do affect soil parameters, how they interact with each other, as well as providing a challenge to study, these challenges inform the nature of the study and provide a means to approximate these various soil conditions through multiple forms of sampling. Comparison of the results provides greater meaning when we understand that percent organic matter content influences CEC values as well as pH values. More frequent cation exchange occurs when more organic matter particles are present, and fluctuations in pH are not as drastic when there are more exchange sites present that a high CEC value provides. From a management standpoint, it is advantageous to know at what pH go-green applications are effective, and in the context of these 5 half-acre plots growing corn, which grows favorably in a pH range of 5.5 to 7, a soil can achieve an optimal pH range for corn. Go-green is applied to soil with a starting pH of less than 8. Regarding CEC, observations regarding relationships to other soil parameters were as expected and included a high value of organic matter as well as a high value of CEC before treatment application, followed by a depletion of these values after harvest, which were also accompanied by a drop in pH. Regardless of pH group, CEC values were found at 8.0, demonstrating a stabilizing effect regardless of pH in plots. Regarding observations of organic matter formation in the study, both the differences in decomposition rates of organic matter in soils less than 8.0 pH suggests that organic matter formation is greater than its decomposition. It appears that the inverse of this trend holds true for soils with a pH of above 8.0, though this needs further testing. And another challenge of measuring changes in organic matter is that it's incremental in 1 to 2% increases per year of the total organic matter in the soil, and so multiple years are required for a complete understanding. Regarding microalgae, composition of populations varies in the soil over several weeks depending on treatment, and numbers are highly influenced by environmental and seasonal factors specific to each region. These results corroborate previous findings and highlight the importance of CEC, organic matter, and pH variations in increased microalgae species diversity through a go-green application. In conclusion, interactions in soil communities play crucial roles in regulating soil quality and processes, and have the potential to be heavily impacted by disturbances such as herbicide and fertilizer application and tillage. Though methods to measure soil quality are highly various, one approach includes the use of microbes to test relationships with soil parameters. And as agricultural operations impact these parameters, these indicators can be used as part of strategies to remediate agricultural soils. Ultimately, application of the trademark microalgae product Go Green demonstrated measurable effects on the previously mentioned soil parameters, the results of which highlight past findings and highlight the importance of such parameters regarding soil quality. This concludes my poster presentation. I'd like to thank Dr. Collin and the Plant Science Department for the chance to present this work. If there are any questions, please feel free to contact me at the email at the bottom right of the page.